A very good morning to all the delegates. Today here we are at the session 2 of day 2 of International Conference on Pharmaceutical Research and Innovations to tackle future healthcare arena. I, Dr. Shilpa Harak, welcome all of you on behalf of Google Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research. Once again, welcome all. It's a pleasure to have two eminent personalities for this session. We have Dr. Praveen Chaudhary Sir, Principal and Professor in Pharmaceutics, has the session chair, and Dr. Bhupinder Singh Bhoop, Professor, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, as the resource person. I take this opportunity now to introduce Dr. Praveen Chaudhary Sir, the session chair for the session 2 of day 2. Sir has completed his B.Pharm from Bharati Vidyapeeth's Pune College of Pharmacy in 1994, M.Pharm in Pharmaceutics from NDMBP's College of Pharmacy, Pune in 1996. After that, he has done MMS in marketing from Bharati Vidyapeeth's IMRD in 1997. He completed his PhD from Bundelkhand, Jhansi University. At present, he is working as a professor and principal at Modern College of Pharmacy, Nigri, Pune. Sir has a total of 24 years of undergraduate experience and 20 years of postgraduate experience. He has an industrial experience of approximately 2 years. He is an approved postgraduate teacher and a PhD guide in pharmaceutical sciences by University of Pune, now Savitribai Phule Pune University. Sir is well known for his knowledge and experience in NAC and NBA and hence serves as a resource person for numerous colleges in and around Maharashtra. Speaking about research, he 15 students have been awarded PhD under his mentorship, and 60 plus students the postgraduate degree of MPharm. Sir has applied for 4 patents and he has 2 patents granted. He has a publication of 156 research papers and 25 review articles. He has presented in more than 15 conferences. He has 6 books to his credit and received various research grants from different granting agencies. I welcome you, sir. For this session, we have the eminent speaker, Dr. Bhupinder Singh Bhoop, who is professor of University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Sir's achievement are no end. He has a glorious achievement list. Sir has been awarded with the Leading Educators of World UK in 2007, the Pharma Personality Award in 2008, Shiksha Ratan Puraskar 2008, Innovative Scientist Award 2012 by CIIPP, QBD and Product Performance Award 2012 and 2013 by AAPS USA. In 2013, he has been awarded QBD Excellence Award by CPHI Asia. He is also awarded Outstanding Scientist Award in 2014 by Select Bio in UK. He has been awarded Pharma QBD Outstanding Performance Award in 2014 by Studies USA. Also, the APTI honored him with the Best Pharmacy Teacher Award in 2014. He was awarded Professor Baichwal Oration Award in 2015 by ICT India. He was awarded Scientist Par Excellence Award 2015 Minitab UK. Udrajit Award 2015 by Evonik Germany. Innovative Healthcare Researcher Award 2016 by WWE. Name in Science Award by EBA Oxford 2016. Professor D.V.S. Jain, Best Scientist Award 2018, Global QPD Excellence Award 2019. Elsewhere, Best Research Paper Awards to 2020 and 21 consecutively, and he has been felicitated by Punjab University Syndicate by 22 times. So, glorious achievement experience. Speaking about his experience, Sir is the chairman at Punjab University, University Institute of Pharmaceutical Science since 2014 to 2017, fellow and member of University Senate and Syndicate from 2012 to 2016, Dean of Faculty of Pharmaceutical Sciences two times 2012 to 2014, and Dean Alumni Relations 2007 to 2011. He is currently the coordinator of UGC Center of Advanced Studies in Pharmaceutical Sciences. He is also founder coordinator of National UGC Center for Excellence in Nano Biomedical Applications, that was in 2011, and he served to a period of 2019. To summarize, Sir has contributed over 38 years of dedicated service towards pharmaceutical education, research, and industry. He is acclaimed globally for his scientific research work, specifically on systemic development of novel and nanostructured drug delivery using QBD paradigms and advanced pharmacokinetic modeling. We welcome you, sir. Welcome, sir. The title of Sir's talk is Quality by Design as an Innovative Tool for Excellence in Pharma Industry, Research and Industry.
Thank you so
Over to you,
much.
sir.
Thank you so much, young lady. I forgot your name. And
It's
Hanji,
okay.
what's your good
Shilpa.
name? Hanji?
Shilpa.
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 Thank you, Shilpa, Dr. Shilpa, for introducing so well and starting the program so dynamically. At the onset, let me thank Professor S.V. Amrutkar and Dr. Prashant Pingle for having me, provided with this opportunity to talk to the young researchers and academicians to share my thoughts and my experiential sojourn uh, on QBD here. Okay.
Sir, can you please share your presentation?
Yeah, I'm doing it. Is it visible? Is it visible?
Uh,
Yes, sir.
yes, sir. Yes, sir.
So, as it has been interested, so I'm going to, although for industry, whenever we tend to train a particular pharmaceutical industrial houses, so we do it for 1 day or 1.5 day or 2 days or 1 week, part-time training program because there are so many facets, so many vistas of quality by design which need to be, known and familiarized and adept in. But within the period of 60 and Dr. Abrutkar and Dr. Prashant have been kind enough to allow me to go beyond the permitted timelines by 10 minutes, 10 to 15 minutes, So I'll try to summarize within the period of around 70 minutes, So it's an innovative tool for attaining excellence in pharmaceutical research and industry because that is the topic of the current conference, international conference. So, my dear young friends, I have structured my talk in this way: little bit about drug product development and genesis of various systematic paradigms, QBD, FBD, that's formulation by design, and some models and applications and the case studies. So I take a drug product from technological perspectives in this way that it's an amalgamation of so many excipients, and of course the drug substance, that's API. The excipients could be functional, high functionality or low functionality excipients. These surface active agents, the emulsifying agent, suspending agent, release controlling polymers, lipids, emulsions, so on, and processes, and some low functionality additives like disintegrants, hardness, imparting agents, so on. Each one, whether high or low functionality excipient, is contributing towards the product performance in their own characteristic manner. And needless to mention, the drug products could be, branded or the generic. Branded are the ones, who are innovators drug product and who hold the valid patent in their name, branded products. A generic drug product is supposed to be the pharmaceutical clone of the branded drug products, which is identical to the reference listed product or branded product or innovator's product in all the traits, and it has to be certified by the federal agencies that it's identical. Primarily, it has to be similar in case of bioavailability. So generic drug products have got phenomenal advantages, particularly the cost advantage. The former president of USA, Barack Obama, used to accentuate on the use of generic medicines to reduce the healthcare cost in the US, and it's valid across the world. Now, in this case, Indian scenario is quite rosy. Very recently during the lockdown pandemic period, we have reiterated the pharmaceutical sector of the country have reiterated its very high position, and we have made the country proud, and we have shown the world that India continues to be the pharmacy capital of the entire world. It's a manufacturing hub for so many countries, accounts for nearly 35 to 45% of the generic needs in US alone. That's phenomenal. Every third drug masters filed in USA is from India. Every third pill taken across the world is from India. And currently it is 1.6 to 1.8 trillion market, US dollar market, which is growing at around, recent statistics tell, it's 22% CAGR, and it is anticipated that in the coming 4 5 years, it will grow further at the rate of 25 to 26% cumulative annual growth rate. That is phenomenal. Let's have three cheers for the Indian pharma scene. Despite the problems of infrastructure, what we call as population, corruption, whatever, you know, Indian pharma's scenario is constantly improving and growing. Although the governments after government, it has not met with, I'm taking this opportunity in, we have not been done proper justice, because we are still continue to be governed by Ministry of Petrochemicals, and chemicals, petroleum and chemicals, which is not befitting to such a big, vast, and performing sector. So DOP is there, Department of Pharmaceuticals is there, but we are constantly looking for a dedicated ministry at the center. That's pharmaceutical ministry. Now, when we compare a branded drug product versus generic product, say left side, we I have taken one example of the branded product, elephant, which has one trunk, four legs, and should be able to carry load, and should be black in color, and should be herbivorous. These are the characteristics of the branded product. Say, for example, I'm taking the example of the elephant. Now look at the generic solution which this man from Kerala has prepared out of a buffalo. Out artificial trunk has been made. So to demonstrate that we have a trunk in our generic product. And four legs, yes, it's demonstrated, and he's demonstrating that the generic product is able to carry load of passenger and cargo, and it has long ears, though artificially made, and it's jet black in color, not only black, jet black in color, and it's certainly 100% herbivorous. And to add additional value, it gives milk too. Could we call this artificially made example as a generic solution? No way. The products need to be identical in holistic essence, not in the individualistic traits, as this guy has very intelligently tried to make. So, a drug delivery system in general, maybe generic or the reference listed generic drug delivery system, it's a specialized formulation which may be, temporal, used for temporal purpose or spatial purpose. Temporal means where the time has been tempered, say controlled release, timed release, or fast release kind of operations. Spatial, where space has been tempered, that's the targeted release product. Another kind of drug delivery systems are, thronging the market in the last 15, 20 years. Those are called as bio-enhanced delivery systems, where neither the time has been changed nor the space has been changed, but the bioavailability has been improved. So in this case, amongst the novel drug delivery systems, nanostructured drug delivery systems is constituting a major pie these days, with significant advantages of uh, surface area per unit volume, and the improved efficacy, and the caution of the possible increased toxicity as well. So the important delivery systems which we have handled while I being at the helm of the affairs in the UC Center for Excellence in Nanobiomedicine in the last 10 years, includes SNEDs, self-nanomulsifying systems, solid nanoparticles, nanostructured lipid carriers, that's NLCs, nanomicelles, nanoliposomes, nanolipopolymexes, quantum dots, dendrimers, graphene, carbon nanotubes, and fullerenes. So, my dear friends, be it novel drug delivery systems or a conventional drug product or a nanostructured system, it has to go through various processes to become a finished product. Say, for example, this raw material is getting granulated and then milled and then dried using fluid bed dryer or compartment dryer, and then again milled and then mixed, say for example using tumble mixer, then finally compressed, over here the rotary tablet compression machine, and then coated. Here it's using XLA quota and then imprinted, and finally you get a imprinted tablet. From the sojourn of this powder raw material to this finished product imprinted tablet, film coated tablet, it has to go through a series of processes. And all these processes are accounting for some kind of variability, some kind of inconsistency possibility is there. So, which is causing the variability in the drug product, and which is causing sometimes failure in the delivering of the goods, delivering of the attributes. So, my dear friends, the complete product quality is governed by various factors. Important ones are governed by these seven M's as dictated using management policies through this Ishikawa fishbone diagrams, which is also called as cause and effect diagram. So the product quality is the effect, and the causes are the men, the scientists and the operators who are operating the machines, and the other cause is machination, all these machines which are used for production and the evaluation, and materials, that's API, the additives, that's excipients, functional, non-functional, all taken together, and including the packages, methodologies, measurement and measurement devices, milieu, that is the environment, and the management policies. All these are accounting for the product quality success or product quality failure. So, my dear friends, the overall variability in the drug product is equal to the variability in API, variability in the excipients, variability in the process, and variability in the packaging, and above all, the variability in the possible interactions between all these. That's interaction between API and excipient one, interaction between API and excipient two, three, N, like interaction between excipient one and excipient two, excipient one and excipient N, interactions between API and process, excipients and process and packaging and API, packaging and excipients. All these permutations and combinations of interactions have to be taken into consideration before we tend to talk about the overall variability in the product. That's very, very important for everybody of us to comprehend, to understand. So variable interaction at times yield weird results. I keep showing this very interesting slide. There is a female zebra, and there is a male donkey, and here is the offspring, which is spotted in a zoo in Australia, and you see the offspring out of this amalgamation may be called as a zonky or a dongra. I leave it to your imagination and to your vocabulary. So, lest this may happen, optimization of drug products is the must. Now, optimization has been the key of development of drug delivery systems too. But question arises, optimization of the drug products has not been taking place in pharmaceutical arena so far. No, optimization has been, a regular phenomenon, but the technique that has been used was one variable at one time, one factor at one time, or changing one single variable at one time. So, which is the technique not coming up to the mark, which is a technique which is half baked. It's also called as trial and error, shotgun approach. So it was marked with numerous, I would again reiterate, numerous pitfalls, numerous disadvantages, shortcomings. I'm going to highlight only a few because of the paucity of time. It is strenuous, it's not economical in terms of money, it's not economical in terms of time, it's not suitable to find errors, what to talk of plugging errors. It results only in just satisfactory solutions, and detailed studies of all variables is not at all possible. It's prohibitive, and you are unable to establish cause and effect relationship. Using this one factor at one time or one variable at one time, and it is just analogous to old story of blind man and an elephant. An elephant was asked to describe by different blind men, so they had different notions depending upon how did they feel about the elephant. The one which was who was feeling the tail part, he said, "Yeah, it's like a rope, it's like a small snake." The one who was feeling the legs, gonna know it's like a tree trunk. The one who was feeling the ears, it's like a fan. The one who was feeling the trunk part, gonna know it's like a python. The one who was feeling the ivory part, he told no, it's like a spear. It's like a bhala. So everybody had different notions. The animal, big animal, remained unexplained before them because they could see only one part of it. That's what was happening in pharmaceutical practice using OFAT or OVAT technique. We had been commenting upon the formulations in a half baked manner. We were not having the holistic view. That's what I shared at you know one of the Makkah of drug delivery, at Sagar. Whenever I went there for evaluation of the PhD thesis, they asked the examiners to talk briefly to the students and young faculty. I told them upfront, you are doing excellent work, but whatever you are doing is half baked because it's you are adopting OFAT or OVAT technique, which is not likely to be yielding the excellent, the best results. So we have to get the best results out of the efforts which the research students are making, and also we have to train them for the futuristic paradigms, which are to be implemented in the drug industry. Drug industry today needs people who are working in the systematic paradigms, not the OFAT technique. So there has been so many recalls from the market. Unfortunately, I have got the statistics till 2017 only. You see, the skyrocketing number of recalls, in terms of thousands, 5,000 recalls nearly in 2016 alone, in USA. Uh, this is exponentially increasing, and primarily it is the cause of poor quality, recalls from the market. And recalls were not restricted to the prescription drugs. They were to over-the-counter drug, cosmetics, medical devices, dietary supplements, that's the nutraceuticals, all were affected. Now, question arises: how can quality be assured? How can recalls and rejects in the market be minimized? Certainly, by reducing variability through rational and systematic optimization, not using OVAT or OFAT, systematic and rational optimization. Fortunately, we have a technique called as design of experiments, which is a rational approach which enables product and process behavior by running a series of well planned experiments. I repeat, well planned experiments, such that maximal information is obtained in minimal experimentation. I reiterate, maximal information, minimal experimentation. So, needless to mention, it's a systematized approach. DOE is a systematized approach. There are so many advantages. Again, time is a deterrent for me. I have highlighted a few of them. It requires fewer experiments. I have already shared maximal information, minimal experimentation, and it yields the best solution. I repeat, the best solution. There can never be a solution better found out than which has been located using DOE. You cannot find a solution better than that. That is the best solution, provided you have implemented DOE correctly. It can trace a problem, and it can plug the problem, rectify a problem. It leads to comprehensive understanding of the formulation system and the process. Today FDA is time and again asking for, product and process understanding, which cannot be diverged unless DOE is implemented. Can simulate the product or process behavior using model set of equations, polynomial equations, and the most important is significant amount of resources are very, very important these days, are saved from being squandered, be it money, be it time, be it experimental efforts, or be it materials. All are getting saved using implementing DOE. So, my dear friends, let it be a bout between OFAT and DOE. Let's see who wins. We carried out in 2005 one such experiment in which we reported to using tenoxicam hydrogel, and here is the flux through human cadaver skin being reported. So there were two variables. One was the carbopol ETD 2000 as the gelling polymer. Another was olic acid as the penetration enhancer. So we first used OFAT technique, and flux through human cadaver skin was plotted versus gelling polymer. You see, the second point gave the maximum flux. So we fixed the polymer concentration at that, and then we varied penetration enhancer concentration, and we got the fifth point as the maximum. Fifth point gave the maximum flux. So keeping these two as the points to be kept in the optimized formulation, the point of intersection of the red beads in the bottom, indicated cost optimum or OFAT optimum. While we carried out one experiment using DOE as well, and the black spot is indicating the true optimum, which was missed altogether, which is quite distant from the cost optimum. So, had we not carried out the studies using DOE, we would have missed the true optimum altogether. Another way of putting forward, the factor A is optimized at the same setting. We tend to optimize factor B, and what we get is a cost optimum or apparent optimum, while the true optimum is real optimum is missed altogether. The scientists have been telling about these techniques in a very different characteristic manner. So I keep sharing Podzak, UK scientist, that dosage form development using trial and error, that is OVAT or OFAT, can be characterized as gambling. If you toss a coin in the air, you may get heads, you may get tails. So if you are adopting OFAT, you may get a good formulation, you may get a bad formulation. It's like gambling, and you always get normally a second class formulation. So this is like that. OVAT is like that, but using DOE, you'll always get the best possible formulation. So in 2003, the US magazine Wall Street Journal shook the entire world. It gave the heading, "The pharmaceutical industry has a little secret. Even as it invents futuristic new drugs, its manufacturing techniques lag far behind those of potato chip and laundry soap makers." You know, that shook the entire pharma world. Probably we are aware or not, the biggest industry today is telecommunications, then is IT, then is oil, and then is pharmaceutical industry. So pharmaceutical industry across the world is a very big industry, and zillions of dollars, and they they were taken aback that Wall Street Journal, very prestigious, very popular, very reputed magazine, is challenging them that the potato chip and laundry soap maker, the nut and bolt industry, the dental implant, pizza industry, etc., they are far ahead. Oil industry is far ahead than the pharmaceutical industry, because we are adopting age old traditional approaches. FDA then started investigating in this manner. Several series of meetings were held, and today we have a concept called as quality by design, implemented by FDA first through ICH, and DOE, design of experiments, is considered to be almost an essential element of QBD. So, as per federal paradigms, the definition of quality by design is systematic approach to design product and process to meet the patient needs at the desired quality levels without relying upon periodic inspection and testing of quality. I repeat, it's a systematic approach to design product and process, understood, to meet the desired patient needs, understood, at the desired quality levels, that we have to maintain the quality levels, but not relying, not depending upon the periodic inspection and testing of the quality. You have to build the quality into the system, not merely test it. So this lady, Janet Woodcock, who reigned the scene of FDA nearly for a decade or so, till the former President Donald Trump, took the reins and laid this lady off to let another, one of his confident take the place of Janet Woodcock. She was very astute lady, and she defined pharmaceutical quality as a function of API, that's drug substance, excipients, process, and packaging. I would like to add to it interactions as well. So it's a pharmaceutical quality is a function of all of these. And USA took the help of GM Duran was the person who was quality expert and who told that quality could be you know uh, built into the system by and to avoid quality crisis rather than testing. You can rely upon building as a team, and uh, into a product. So he was not properly listened to in US. He moved to Japan, and the first company which followed GM Duran's philosophy, that's QBD, without telling it QBD, was a Toyota company. So Toyota company, and then there was no looking back. He was consulted to so many other companies. He was guiding them on building quality into the system. Then his services were recalled by USA. He was requested to come back to steer the pharma world as well. So he obliged FDA and guided to implement what's called as quality by design paradigms these days. So, my dear friends, Duran's philosophy of QBD is quality by design or quality by planning, not merely by chance. Earlier it was quality by, you know, chance, quality by probability. Now it's quality by planning, requisite planning and execution. So, what is quality? If you look into the dictionary meaning, quality is building excellence in a trade, and excellence is building quality in a trade. In other words, quality and excellence go hand in hand. So, design. What is design? Design is conscious intent. It's an intent. In day-to-day life भी हम कहते हैं, this guy doesn't seem to have nice designs. No? Uh, th- this guy seems to have nefarious designs. Design means plans, intention. So, so design over here is not the experimental design in quality by design. It's the intent, quality by planning, quality by intent. So it's an intent to deliver the quality of a product or process to satiate the requirements of the customers. Customers for us are the patients. So doing it right from the start. I would request my our audience, to be little patient because I'm going to show them nearly 180 slides and within this period of 70 minutes. So doing it right from the start. It's all about patience, understanding the patient needs, designing and developing products meeting those patient needs, designing and developing the manufacturing process capable of delivering the products meeting these patient needs. Patient needs. So QBD settings even let you go beyond the requirements of the customer, that's the patient. So you can go beyond. You can just see the QBD expectations are the voice of the process, while the customer expectations are within the lower specification limits and upper specification limits. That's what I keep sharing to industry. QBD gives you good return on investment, enhanced knowledge sharing, improved time to market. Industry understands this because they are competing. So if anybody is able to launch the product earlier, they are going to be having extra advantage. Reduced cost because the recalls will be less, rejects will be less. The overall cost of the product will be much more economical. Generic skepticism would be less. People will be more confident on generics today. If I have to buy a product for my, say, grand son or granddaughter, and my daughter will be telling, okay, Papa, please buy the best product in the market, you know don't compromise. So I will not be going in for a generic substitute which is costing maybe one fourth or one fifth, because I'm little doubtful about the quality. So with QBD in place, that doubt, that skepticism will go away. Minimize post approval changes. It will be much more cost effective as well. So it also provides regulatory flexibility. That's what industry always looks for. So in terms of design space, so it's a win-win situation not only for industry and patients, but for regulatory agencies as well. Look at the 2008 filing left side and 2014 filing post QBD. Look at the volume of filing has drastically been reduced. And earlier left side, data was rich. So much of data was submitted and information was poor. Today with the QBD in place, everything is systematized, and you can get any piece of information at the touch of a single button and get a pertinent printout of the same as well. So quality by testing, the traditional approach, or quality by inspection, was black box all across, and end testing was must. So particularly the products which were getting tested for bioavailability, people used to have nightmarish experience, kal kya hoga. But using quality by design and testing is optional, and everything is transparent. No ambiguity is required. Kal kya hoga is gone, because kal kya hoga is known to everybody using QBD, that kal acha hoga, because we have built the quality into the system, not we are merely testing it. The level of confidence, level of credence has drastically been changed. So quality by testing approach was testing intensive. QBD approach is taking design and testing together. So QBT is like chaotic traffic of our New Delhi, while QBD is the traffic regulated of Los Angeles. I clicked this photograph in 2004 or 5. You look around, now things may have changed for the betterment. How regulated the traffic is. QBT is like diving in an ocean full of uncertainty, while QBD is driving on that ocean, over well carved path. QBT is like, tip of the iceberg. Big iceberg is submerged in water, while QBD is tip as well as the submerged iceberg taken together. QBT tries to fit the same attire to different individuals, but fails to do so, while QBD lets it happen. So QBT banks upon knowledge exchange from one person to another, one company to another, while QBD banks upon not knowledge exchange, but knowledge integration from various siloed departments, from segregated departments like QA, QC, analysis, pharma manufacturing, IPR, DRA, and of course the formulation R&D taken together. So there is another concept which is coming up in the federal paradigms is QBR, which is called as question-based review. So each product has to be, examined by the federal agencies through question-based reviews. They are asking the scientists about certain questions, because if a product is faulty, hundreds of thousands of the patients who are taking that faulty product, their life is likely to be at peril. So FDA is more concerned about the patients. So they tend to ask a series of questions from the scientists. How well do they know their formulation, or how well do they know their process? So they can tell about the product and process understanding only if they have implemented QBD. So QBD is in tandem with the QBR. So QBD was implemented in 2019 to be, implemented in totality by January 2013. But 2009 to 2013, 4-year period was given to them. But industry raised so many hues and cries that it should be postponed for another 2 years, but it was not done by FDA. So history is witness, January 1, 2013 onwards, all products reaching USA have to be QBD compliant, particularly the generic ones. The situation is still alarming, with many industries, but I must share that many of the leading pharma industrial houses of our country are QBD compliant today. Genovia was the first product which was QBD compliant. Since then, there have been, exponential rise in the QBD compliant products. This is worth clicking slides. That is the various steps involved in QBD. First, you have to select the quality target product profile. That is the objective. Then you have to delineate some critical quality attributes out of QTPP, quality target product profile. Then you have to select some critical material attributes and critical process parameters, which are the input variables, and you have to link these with the output variables, that is critical quality attributes. And that linkage, that association, that understanding would help us to develop a design space within which the formulation would behave, and would the specifications tend to remain within. And the entire process from QTPP to till design space is the milestone, the roadmap is earmarked in a strategy called as control strategy, where various controls are earlined, earmarked. And this control strategy leads to continuous improvement even after the commercialization of the drug products. QTPP, CQAs, CMAs, CPPs, linking of CQAs and CMAs, and then postulation of design space, postulation of control strategy leading to continuous improvement, and the cycle goes on. So QTPP. So QTPP is what is route of administration, what is dosage strength, what are the containers closure system, what are the drug delivery criteria like drug release, stability, etc. What are other attributes affecting the pharmacokinetic performance like drug release or dissolution, or in case of inhalable system, aerodynamics, so on and so forth. One example of the tablet dosage form, where the QTPP is dosage form, route of administration, tablet weight, assay impurities, etc. Out of these, important parameters are chosen as CQAs. For clinical effectiveness, it's assay, content uniformity for assuring the consistency, and dissolution. For safety, it is impurity. So out of QTPP, some parameters are chosen, which will be studied further extensively. These are called as critical quality attributes, and these are physical, chemical, biological, or microbiological properties which should remain within certain range. So I keep asking the scientists whether critical quality attributes should be for company profit. Answer is no. Operator safety. Answer is no. Environmental safety, no. It's only for patient benefits. Patient benefits, patient benefits, and patient benefits. That's what we need to look at for deciding critical quality attributes. And critical material attributes are with respect to the API, excipients, or packaging materials. Critical process parameters are with respect to the process factors like rotor speed and granulation time, etc., etc. So, product and process understanding comes through the QBD in nutshell. You look at the outermost shell. So, after implementing, this is out of the FDA site. Product understanding, outermost shell tells process understanding. These two are the twin achievements of implementing QBD. So when FDA is asking about QBR, question-based reviews, so product and process understanding would be diverged after we have implemented QBD and leading to product and process understanding. If you understand the product and process, you will be able to answer question-based reviews. And we need not worry. There are guidances to guide us on Q8, Q9, Q10, Q11, and Q12 very recently, and PAT guidelines, PAT guidelines, that's process analytical technology guidelines. All these are working in tandem with the quality by design principles. And today, those ICH guidelines have been, endorsed by all the leading regulatory agencies, WHO, FDA, EMEA, Europe, that's MCC, South Africa, Health Canada, SFDA, China, MHRA, UK, PMDA, Japan, Australian, TGA. And let me share, Indian Drug Control, CDSCO has carried out a series of meetings, in 2018 and 19, and the report has been submitted in 2020 through CDSCO to Parliament. But I learned, this is going to happen in the next Parliament meeting, where the Drugs and Cosmetics Act and Drugs and Cosmetics rules will be changed for the drug products, and where the elements of QBD are also implemented. I was also fortunately member of that committee, and so was late Professor Mukesh Gohil from Ahmedabad, and Professor Saranjit is also one of the members from NIPER. Besides so many, people, Dr. Romi Singh, Dr. Pradeep Mattu, etc., from industry and drug regulatory agencies. When QBD has been the talk of the town, we propose another term in QBD in product development somewhere in 2015. That's formulation by design. Formulation by design involves the basic principles of drug product development, QBD based understanding, quality risk management. I'll just briefly explain that, and design of experiments. This was our first review, out of a formulation by design. And then now today, every now and then, a formulation by design is being used. This is out of the advertisement of Ford Delta, a leading formulation by design company, which gives the heading as formulation by design. So it's the best tool to match the innovator product's performance. OFAT technique may hit here and there, while formulation by design would always be hitting the bullseye. So there are so many advantages: best solution, robust, holistic, systematic. And as per formulation by design, drug formulation is considered to be a system where transfer function is taking place from the input, that's X, to Y, while there are some variables which are uncontrollable, which are called as U. And if the outcome is highly predictable, it's called as white box. If it at all is not all predictable, it's black box. If it is moderately predictable, it's called as gray box. Using formulation by design, it's always white box or nearly white box status, while using the traditional approaches of quality by testing or OFAT, it's black box or nearly black box status. So it tells you what's happening inside. So there are some definitions which need to be made aware of. I can pass on the manuscripts, the publications which are highly sought after, to the organizers, to Dr. Amrutkar and Dr. Prashant, for those aspirants who wish to know about nitty-gritty of formulation by design. What are objectives? That's QTPP, variables, what are factors, critical material attributes, critical process parameter responses, critical quality attributes, values assigned to the CMAs, that's levels, and what are experimental runs, that's the trials as per experimental design, what is the factor space, what is the knowledge space, what is the design space, so on and so forth. So variables could be CQAs, CMAs, CPPs. CQAs are the dependent variables, while CMAs are and CPPs are independent ones. So coding is absolutely necessary. So here one example I have shown out of one research practices, where polaxamer has been used in different amounts. That's 25, 50, 75. The low level is coded as -1. High level 75 is coded as +1. The average of the same, that's 0, is coded as the average of these both of these, that's 50. Similarly, PVP K25, the other excipient, was used in 50, 100 as low level -1 and +1. high level, and the average, that 75 has been coded as 0. And when the response variable, that CQAs, versus one of the input variables, show parallelism, there's no interaction. And when there's no interaction, there's no advantage of DOE over OFAC. But when there is interaction, which is happening very frequently, we, being the humble students of pharmacology, we know most drugs, when given simultaneously, they do not behave A plus B is equal to A plus B. The overall response is either higher or lower than the cumulative response, leading to synergism or antagonism. So if it is divergent, it's called as synergism. If it is convergent, it's called as antagonism. And 3D response surface, which is a plot between y and x1, x2. X1, x2 are two variables here: completol and tweenity versus particle size, which gets generated. A 2D slice, transverse slice of the same, or top view of the same, gives you a 2D contour plot. The details can be diverged. I beg your pardon, 1 hour lecture or 70-minute lecture cannot make you, proficient in QBD. It can just sensitize you. It can provide impetus to you to go in for QBD or FBD. So I can provide you with enough literature. Each article is being sold by different journals at $180, $200, $220. But we, as the corresponding authors, can easily provide you with a copy for academic purpose. So it's a five-step methodology. I'll cut it short in a few minutes. First step, QTPP and CQAs. Second, CMAs and CPPs are defined. The technique is called as factor screening or risk management. Third, you will be carrying out experimentation as guided by experimental designs. Fourth, you'll be developing modelization, and you'll be developing design and control spaces, and you'll be validating as well. Fifth step is taken up in industrial milieu, and you are carrying out pilot plant scale up studies and production and commercial level batch is prepared. Step one, QTPP and CQS are defined. Already we have discussed. Step two, you tend to decide the highest priority variables. So this is based upon the Pareto principle. That is, 80% results come from 20% of the causes. Say 80% of the wealth of the world is owned by 20% of the rich. 80% of the fatalities across the world are due to 20% of the diseases. And in marketing its store, 80% of your products are being consumed by 20% of your consumers. You have to, highlight those 20% of the consumers, not the 80% of the consumers who are just accounting for 20% of your. So in DOE and FBD2, we are to delineate that 20% of the variables which are causing 80% of the variability, and for that, factor screening is carried out. So many, input variables, and we are left with so less number of critical material attributes and critical process parameters. So it's like separating rice from rice husk. And if an input variable is a factor, influenceable factor, and you have been able to identify it, it's going to yield the correct optimum. But if it is a factor you are not been able to identify, God is the savior. It will always be leading to erroneous outcome. If it is not a factor, you have not been able to identify, well, it's a desirable situation. But if it is not a factor, you have unnecessarily been able to identify it as a factor, so it will lead higher inputs, higher cost. Two techniques, half normal plots and Pareto charts, are used to carry out screening exercise. Besides the screening, another approach which is thronging the industry is risk assessment or risk management. And in fact, I have been training so many pharmaceutical industry in India and beyond India. Many pharmaceutical industries like Sanofi, Aventis, and Mylan had been adopting risk management since decades. You know, and it was not very difficult for them to adopt QBD because part of it they were already implementing. So it's like you are trying to walk amongst the tight rope, and which is amongst so many possible risks and dangers. It's not merely training you to walk on rope amongst dangers. It provides you with cushion, quality risk management or risk assessment. This is the risk estimation metrics. The green squares or rectangles are to be ignored. Red ones are to be taken care of, while in between you can take a call. Another approach is failure mode effect analysis, which is adopted in various pharmaceutical industry. So this dental surgeon, to break the monotony, tells your teeth look great, but it looks like that we need to extract your tongue first because it's coming the way of your teeth. So risk assessment helps you to extract the tongue without taking out the tongue. So it requires uh, brainstorming among team members, and not only lab scale, pilot scale, exhibit scale. It's valid uh, till that. It goes even beyond post commercial launch as well. That's still filing. So FBD or QBD is applicable throughout the life cycle of the product. So during prioritization, during experimental planning, during experimentation, and during implementation, FBD cycle goes on and on and on. So step three is experimental guided experimentation and analysis using DOE. Experimental design is very very important component of the same, which is a strategy to set up experiments. हम तेरे बिन अब रह नहीं सकते. This is the theme song which was being played not for the entertainment of the audience, but to demonstrate that many a times this kind of melodious music is accompanied with the noise. As I repeat, the song as well as the noise. So the noise is not able to let the music being enjoyed. So that's happening in day-to-day situation as well. So we have signals, we have noises. As this lady, who has come to the park after quarreling with her husband, you know, is finding the music which is being loved by the other guy as noise. For her, the music is a noise, while the guy who's trying to listen to the music as the signal, for her, it's a noise. So it depends what is signal, what is noise for you. So in day-to-day life, the desirable situations come which are like signals, undesirable situations are like noises, but you have to accept them such. In the science, you can increase or modulate signal to noise ratio by adopting experimental designs. So you can decouple the signal from noise uh, using coconut water, using experimental design. So experimental design is an technique which is helpful in controlling signal to noise ratio and maximize the same. So another important aspect is power of an experimental design. To illustrate the same, here, I repeat, here is the golf ball struck, and the golf ball was located to be here. Now, what is the probability that golf ball will be spotted? It will depend upon the length of the grass. So, if the length of the grass is twice that of the ball, there is 15% possibility that you may be able to spot that ball. So here, the ball is the signal, noise is the grass. So if the ball diameter and grass length are same, then 45% possibility is that you will be able to, you know, hit the bullseye. You'll be able to hit the the ball amongst the grass. And if it is half grass is half that of the ball, then the possibility of it being spotted is as high as 96%. So experimental designs could be screening design, response surface designs. I'll just name. Time does not permit me to go into details. Factorial design, central composite design, box blanket design, simplex design, optimal design, fractional factorial design, Taguchi design, placket Burma design. These are the important designs which are used in pharmaceutical factories. The first ones are used for optimization, while the last four are preferred for factor screening. So this is how they are diagrammatically represented. Time does not permit me. Those articles will educate you. As central composite designs, we are very fond of CCD, which is consisting of three parts: the factorial points or the vertices, central points 1, 2, 3, 4, 5. The central replicates and axial points. The star points, 4 points. So 4 plus 5 plus 4. 13 runs are needed for two factor design. We find it has an excellent design for two factors. And Sir Ronald Fisher, the father of experimental design, used to tell that simple designs should be preferred. Advantages, disadvantages of various designs. I'll pass out the article where all these advantages are mentioned. Step four is, you know, control and design spaces and optimum formulation. For that, graphical approach is adopted. That's overlay method. Mathematical approach like desirability function is adopted, or artificial neural networks could be adopted for that purpose. And we are living in an era of validation. So diagnostic plots, validation plots are carried out. One such plots is residual plots, that's observed minus predicted. Plots should not show any kind of trends. A is desirable, while B, C, and D are the undesirable trends. So many times, more than one approach is adopted to find the optimum search. Like this, the apex is the desirable point. The This red point is the starting point. So when you tried, you missed the apex point, and using linear approaches. Then you reached close. Then again you adopted the second linear approach. You missed it. But when after reaching that you adopted second order model non-linear approach, you missed. You hit the bullseye. You got the best formulation. So many times, linear and non-linear, first order and second order, techniques have to be followed correctly. Say, for example, going to Nasik, I may have to first fly to Bombay, and then move to Nasik via road. So first by air and then by road. Two approaches: first order and second order approaches. So we are having explorable space within that knowledge space, design space, and we should move within that control space. So control space within which we should remain. Design space, if you go out, it will be considered as a changed formulation. And I have just tried to explain that if you have a farmhouse, you'll remain within the farmhouse. But once in blue moon, you can come out of that farmhouse and go within that picturesque lawns, and you will not be called. You'll not be called as you know as a changed formulation. But if you go beyond the picturesque lawn, people may call that you have trespassed your boundaries. That's it's a changed formulation. So fifth is production at the industrial scale. And design space is very important aspect. You have to find the appropriate tools to search for, as this dental surgeon is doing. And for that, the lab scale uh, design space has to be translated to commercial scale. For that, the equipment has to be dynamically and kinetically and geometrically similar. And to find out design space, you have to tighten the operating space and lead through the control space. So you normally remain within control space, and but you cannot go uh, beyond a design space. So lab scale design space has no regulatory importance. So is pilot scale design space. It is the commercial scale design space which is required. So there are software to help us out. Design expert, Umatrix, Camo, Minitab, etc. I keep recommending Design Expert, Minitab, and GMP for the purpose. GMP is a little costlier. Design Expert training is costlier. So is software. Minitab is a general purpose software which provides excellent facilities for DOE and quality risk management and factor screening as well. So it provides other statistical information as well, and it's affordable as well. So here is the best formulation, and it's like celebration time in Punjab. Chak de fatte balle balle. Now here, and last, I'm going to conclude. Before concluding, I'm just cautioning you. In 2011-12, people told that science is almost dead, nicely confined to design space, everybody is talking about. But where is the science? So question arises, is design space the only objective or the outcome of QBD exercises? No, not at all. It is the product and process understanding through science and risk based approaches. Now, Albert Einstein told, any fool can know. The point is to understand. So, to make my point across, I have brought some TV clips.
He's going to order samosas. I know your heart only too well, so I make Kellogg's heart to heart toast every day. He's going to put his arm around me. After all, who knows his heart better than me? Kellogg's heart to heart toast. Jack, lunch में घर आओगे बेटा? लाइब्रेरी जाना है?
नहीं माँ, लाइब्रेरी जाना है।
और डिनर?
ग्रुप स्टडीज है माँ।
चाय तो पीता जा बेटा।
हाँ, तुम्हारी चाय हाँ।
Now, in both of these ads, TV ads, Kellogg's Heart to Heart or Group 1 Tea, I'm not going to get any kind of commission from either of them. But you see, the lady was able to predict the next move of her husband, and the mother was able to predict what is her son going to answer in response to the questions. How? Why? Because the lady, both the ladies were, able to understand their husband and son respectively. So where there is understanding, there is predictability. That is the outcome. And where there is predictability, there are no errors. If you understand, if you predict your process and product very well, where can they lead to any kind of errors? Even if there is an error, you can easily plug them because you understand them, you can model it. So that is the purpose for product and process understanding to be developed by the federal agencies. So prior knowledge, prior patents, prior publications lead to prior understanding. Product understanding, for that, IVIVC, product knowledge, pharmacokinetic simulations, etc., is required for process understanding, process engineering, process capability indices. For that, I keep recommending to pharmaceutical industry that it's highly advisable to include one chemical engineer in your team, particularly in the plants. That will make a good combination. Not all pharmacists would make a good synergistic combination. One chemical engineering would certainly add because they are trained in process understanding. So this is out of on one article which we published very recently on QBD based drug delivery development, nanostructured systems, you see exponential use of QBD has been reported, on various uh, development of various nanostructured and novel drug systems, and it's applicable to all kinds of systems, nanoemulsions, polymeric nanoparticles, carbon nanotubes, nanocrystals, nanosuspension, nanovesicular systems, and you see all kinds of experimental designs are used. CCD is very common, FD is very common, box Benkel design is very common, mixture designs are very common, while rest are less commonly used. Now, at this stage, you know, question arises: QBD is an option, it's a force, coercion, or it's a benediction. Benediction means blessing. So I presume it's neither an option nor a coercion. It's a benediction. It's a blessing which gives you pharma excellence and regulatory compliance both two in one, and you can remain relaxed and meditative in such an environment. So API development today, QBD is not mainly for formulation development. API development, yes. Analytical development, yes. Analytical quality by design, we have nearly 60 publications, research publications on analytical quality by design. It's an emerging area. ICHQ 14 guidelines are coming up, which will be dealing with analytical quality by design. Formulation development, we have discussed, dissolution testing, manufacturing process, biochemical studies, stability studies. For all these, QBD is applicable. And stance and stand is very much valid. The sad kind of stance is not going to work. The pleasing stand, pleasing frame of mind is going to work. Chalta hai attitude is not the option. Chalna hai attitude should be the attitude. And Jugaad technology is not going to work. Juran technology is certainly going to work. Together, everyone achieves more. It's a team which is needed for QBD. And there are some cautions and tips. Again, time does not permit me, but you should not go beyond the limits within which you have carried out the analysis. Extrapolation is not required. And when the whole world is knowing your problem, the wind is at your back. Act now. Some 410 publications and lectures and on majorly on QBD, and some highly sought after articles on QBD, and which I'll be ready to part with depending upon your requirement. Some 10 books on biomedicine, some tech transfers to industry, and the industrial houses with which I have been able to work in the last, 2 decades or so, including from Taiwan, Bangladesh, Dubai, and Canada. And this is the CEO of Statis company, Pat Whitcomb, who came to my hotel in Mumbai 2013 to seek my help in QBD exercises. And here is our meeting with the FDA reviewers, Dr. Vilayat Saeed and Dr. Lane Constantins and Dr. Albinus D'Souza, and with Professor Saranjeet and me. Uh, we learned a lot from each other during these interviews. Dr. Ijaz Hussain, when QBD was implemented, he was the deputy, FDA director, deputy director of FDA, and he has been a good friend on implementing QBD in pharma development. CPHI Asia, my senior, respected senior, whom we lost last year, Professor Mukesh Goel, I getting awarded from his hands, and many other awards. Recently, 2019, China, they honored me with QBD Excellence Award, and my students, Dr. Rishi Kapil, Dr. Kesar Raza, Dr. Sarwar Beg, Dr. Rajneet Kaur Khurana, they were awarded with QBD awards at various international platforms, national and international platforms, and uh, UIPS has been maintaining consistently second rank. This is 2016, and in research we are primarily number one. And thanks to the predecessors like Professor Kishan Singh ji, who has been our mentor since decades, again whom we lost unfortunately in March 2020. Attitude determines altitude, hence we need to transform our attitude. A journey of 1,000 miles has to start with a single leap. We need to fly at the wing pace to be at the global standards and stand apart in the world of mediocres. Thank you so much. Then the mother India would stand prominently on the world map. Jai Hind. Thank you so much. And this is my team. Normally we work in unison with that of the team of Professor Katare. This is our team of postdocs, research scholars, and post. This is the green campus where we live. Thank you so much. I hope Professor Praveen Chaudhary ji, Principal Praveen Chaudhary ji, the chairperson of this, program, finds I not going much beyond the time limits. And thanks for bearing with me. Professor Praveen Chaudhary has been my young friend whom I have learned a lot, during various frequent encounters which we had during APTI meetings, during NBA meetings, and during PCI meetings. Thank you so much for all for patient listening. And if time permits, you can ask questions. If time does not permit me, you can email me question. I'll respond at my earliest convenience. Thank you so much.
Thank you, sir. There is one question in the stream. I'll just highlight it. Uh, Mr. Someshwar Mankar is asking whether there are any free QBD softwares available or only paid versions.
Free, yeah, demo software are available. There's nothing like free lunch in this world, Dr. Sumeshwar. Uh, but I tell you, briefly. In truth, we started implementing QBD into 1995. So we started investigating whether any free software are available, and even in this exchange to our own software. But we failed to do so. Then we developed our own software that's FacTalk using Fortran language and C++ language, and we started working in that for 5 6 years. But that was not very versatile platform. Then we started buying design expert software. Now for the last almost 10 years, design experts software has been provided gratis to me, perpetual license, and 10 of my users, that's a single user annual license. Ours is the only university across the world where they have been providing free license in response to the extraordinary work, exemplary work on QBD based drug delivery system as per their standards. But it's very, very rare. But demo version, 30 days, 45 days versions are easily available in the market. Yes.
Yes, I'll request Dr. P.D. Chaudhary, sir, to give his concluding remarks. Please, sir.
Yes, Shilpa madam. Uh, it's my pleasure and honor to chair a station of such a stalwart in pharmacy education field, Dr. Bhupinder Singh Bhoop sir. Thank you, Dr. Bhupinder Singh Bhoop sir, for updating all the participants on QBD as an innovative tool for excellence in pharmaceuticals. You made this talk so interesting by giving very simple example. Overall, I would say this talk is really informative and useful for the young researcher. And I really appreciate the way you have simplified your talk with the various examples. Being an academician, I think we all are updated with the recent trends in the industry. We can percolate the same to our students, train them through projects, and give industry ready graduates and postgraduates to cater the demands of Indian pharmaceutical industry. In this context, Dr. Bhupinder Singh Bhoop has enlightened us with the knowledge of generic versus branded medicine, Indian and world scenario of pharmaceutical market, nanostructure drug delivery system, pharmaceutical product development through optimization, and optimization of pharmaceutical product by design of experimentation. He also shared the Zuran's philosophy of QBD, quality by planning and not by chance, doing a right from the beginning, and how this implementation of QBD is the win-win situation in the pharmaceutical industry. He also enlightened us with the concept of quality-based reviews, steps involved in QBDs, quality target product profile, and critical quality attributes, critical material attributes, and their importance. ICH guidelines, concept of formulation by design, hurdles involved are hurdles to cross for formulation by designs. In FBD in product and process development, risk assessment in FDPs, and many more points he covered with various experimental designs and the software used for these experimental designs. Overall, he updated all participants with the current knowledge on QBD and provided participants with the ideas about how this knowledge can be implemented in their research projects. I'm very much sure all the participants must have benefited with this his talk. To conclude, as per the new ICS guidelines, QBT refers to a holistic approach towards drug development, and it has become the answer to assist both industry and FDA to move towards a more systematic, scientific, risk-based, and proactive approach to the pharmaceutical product development. QBD offers several benefits for industry, like better understanding of the product, manufacturing process, less batch failure, more efficient and effective control of change and return of investment or cost saving, basically building a quality in and not testing it. So design experts, risk assessment, process analytical technology, that is PAT, are the tools that may be used in the QBD process whenever it is appropriate. Thank you, Dr. Bhupinder Singh Bhoop, sir. Once again, I want to thank you for enlightening us via such a wonderful talk on QBD and its importance in pharmaceuticals. I wish this international e-conference will be the grand success. At last, I would like to thank the organizer, Gokule Education Society, Sir Dr. M.S. Gosavi, of College of Pharmaceutical Education and Research, Nasik, Dr. S.V. Amrutkar, Principal, and Dr. Pingle, Coordinator of this e-conference. Thank you, sir, for giving me this opportunity and share my views on this occasion. Thanks a lot. Thank you, Dr. Bhoop, sir.
Excellent, excellent analysis, and thanks for the pearls of wisdom, Dr. Praveen, as usual. Really, really, really impressed with your analysis and your knowledge of the concept and of the pharmaceutical technology. Thank you so much. Thank you.
Thank you, sir. Thank you.
And thank you so much, the organizers of Gokhale Educational Society, Dr. Amrutkar, and particularly Dr. Prashant and Dr. Shilpa for nice anchoring. Thank you so much. God bless. All the best. All the best. Bye.
Thank you, Dr. P.D. Chaudhary, sir. I request our principal, sir, to present, sir, with a certificate for being the session chair.
Thank you. Thank you, Dr. Sunil.
I also request him to present the certificate to I also request him to present the certificate to Dr. Gupindra Singh Bhoop, sir, for kindly accepting to be the eminent speaker. Thank you, sir. Shall we end the session, sir? We'll go
Yes,
offline.
yes, yes.
Yes.
Thank you so much.